The format for the rest of the day will have two sessions. The first session roughly at the body scale, and the second session roughly at the population scale. In each session, I will introduce the speakers as a block. They will, one after the other, give their talks, and then we'll convene all of them together and have Q&A. So please ask your questions as the speakers are presenting, but the questions I will relay to the speakers, after we convene as a panel, after the group of 4 or the group of 3. As well, we'll have a little bit of history trivia. To kick off the session on the body, have in the back of your mind, just reflect, when was the first ECG used? When was the first full body CT scanner available? And then very topically, in terms of the needs for addressing COVID, as a very important technique to measure the oxygenation in blood, when was the first pulse ox actually used in clinical care? We'll answer those questions at the end of the speaker while we give them the chance to convene to the collective panel. So it's my pleasure now to introduce our 4 speakers for this session. I'd like to introduce Lee Gehrke, Giovanni Traverso, Canan Dagdeviren, and Michael Cima. Lee will kick it off talking about rapid antigen diagnostics for emergent pathogens. Giovanni will talk about gastrointestinal device development. Canan will talk about electronic textile conformable suit, E-TeCS. And Michael will talk about magnetic resonance relaxometer for improving clinical outcomes in hemodialysis. So please ask your questions as the speakers go, and I will shepherd them along to the speakers at the panel at the end. With this, Lee, I turn it over to you. Thank you.
Good afternoon, everyone. I'm very happy to be here to tell you a bit about our work on rapid antigen diagnostics for emerging pathogens. This technology is very dependent on biomaterials at the nanoscale, and all this is critical for making these kinds of tests. So this is an image that is all too familiar to all of us these days. It is a testing tent where individuals are being diagnosed or surveillance is being done for COVID disease. And the need for rapid diagnostics to detect these pathogens has become acutely a problem during the COVID era. So if we start at the beginning and ask, what are the creatures who are spreading these viruses, and how are they getting in contact with human beings to cause these pandemics? And so the first one that I'll mention is the common mosquito, which all of us are familiar with. In the Northeast, the mosquito carries, for example, West Nile virus, which was a problem a few years ago, but fortunately didn't grow into a serious epidemic or pandemic. But nonetheless, mosquitoes carry a lot of viruses. Another one is ticks. Ticks are becoming an increasing problem, again, here in the Northeast and also in the Midwest. Ticks carry an emerging virus that's called Powassan virus. Powassan is dangerous because it causes a serious encephalitis. And so this is an example of one we have to watch. The third example of animals that carry viruses is the mouse. For people who might live in the Southwest, this is the creature that spreads hantavirus, another serious pathogen that could be emerging into a bigger problem. And then finally, bats. And bats are being associated with more and more serious viruses. So why does this all happen? How do we go from these viruses that normally live in what we call a sylvatic existence? So they're in arboreal regions, in jungles, and usually circulate in animal populations and aren't much of a problem for humans. But if there is what we call a spillover event, in other words, these viruses or their carriers, the animals, come into contact with humans, and this can be in a number of different settings with environmental disasters of fires or over farming and population density, these animals come in contact with humans and there's a spillover event, and then that virus is carried over into urban areas where the virus can cause a pandemic, as we're seeing with COVID. So in thinking about these viruses, there are a number of different kinds of rapid tests that have been developed. And I want to review the three kinds to distinguish them, and also to point out the kind of diagnostics that we're working on. So the first of these tests is a so-called PCR test. This is a test that detects the viral RNA or nucleic acid. The second test is a so-called serological test. So this is a test that detects antibodies in the blood and is important in telling us about the history of the disease, when people were infected, how long ago, and whether or not they're producing antibodies. And then the third test is the one that we're working on, and this is called the direct antigen rapid test. And in this test, it's a non-amplifying technology as compared to the PCR tests, but we're detecting directly the proteins that are on the virus. So, and I'm pointing out here, the pinkish structures are the viral spike protein, and we directly detect those to determine whether or not the virus is present. So these tests are looking much like a pregnancy test to give you a visual of it. And the goal has been to develop tests that are low cost, don't require a lot of expertise to use, and can be scaled up for manufacturing. So this is what a pregnancy test would look like. A fluid like urine is applied to a sample pad, and then it flows along by capillary action. And if there are two bands that are present, it's a positive test. If there's only one band at the positive control, a single band means it's a negative test. And importantly in here, as part of the construction, is this area called the conjugate pad. And in it, it contains these round structures which depict nanospheres or gold nanoparticles that are conjugated to antibodies, as indicated by this Y here. And so in the case of pregnancy test, a test is dipped into or has urine applied to it. And if there's only a single band, that means the individual is not pregnant. If you have two bands, then there is a positive test. But here we're not detecting pregnancy, but rather viruses. So instead of using and detecting hormones, we're detecting virus that's present in swabs or sputum or saliva and so on. And again, the second band here indicates that it's a positive test. And if we look at the test line a little bit closer, that would be this line here. What's going on is that there is one antibody that's bound and mobilized to the paper. It traps the virus itself by interacting with the proteins. And then there's a second antibody that's bound to a gold nanosphere. And these collect at the line here, the test line, and that's why you can see a pinkish line because of these collection of gold nanospheres. So in thinking about emerging viruses or families of viruses, here's a problem that we have, and that is that the proteins of related viruses are very similar. So how can we tell them apart? How can we tell which is the virus that's causing an infection? And here's a list of some of these viruses that are very similar: West Nile, dengue, Zika, Japanese encephalitis, powassan. These are all from the same family, as are SARS-CoV-1 and SARS-CoV-2. So how do we think about being able to distinguish these kinds of viruses? And one answer to that is that we use more than one nanoparticle. And what I'm showing here is a pinkish nanosphere. And now we're also using nanostars, gold nanostars, that give us a second dimension in being able to detect and distinguish these viruses. So now that gives us two different types of particles to use, again, giving us another dimension of separation and identification. So here's one type of detection using gold nanospheres, which are pinkish, and then we have gold nanostars that are blue. So that allows us to have methods for separating these different pathogens, and even in a test tube, you can tell them apart. These are the nanospheres, these are the nanostars. If you look at them under the EM, these are the nanostars here, and the nanospheres are over here. This is about 40 nanometers or so in diameter. So here's what the tests look like. This is like the pregnancy test. So this sample was applied down here, and then it flows up toward the top. And you can see that we're getting spots in different areas. And so there are two dimensions here where we're seeing a spot, and secondly, what color it is. So here's a blue spot here, a pink spot here. And now by using a little bit of image processing and red, green, blue RGB analysis, we can analyze these even on a mobile phone and do a principal component analysis. And the data right here are showing the ability to distinguish dengue 1, dengue 2, dengue 3, and dengue 4. These proteins are extremely similar, but by using image analysis and principal component analysis, we can clearly distinguish these clusters and be able to tell these different viruses apart. Another one that we've done is recently is that we can distinguish SARS-1, the virus that appeared in the early 2000s, from the pandemic strain of SARS-2, again with using multicolored nanoparticles. And you can see these different colored nanoparticles here. This one's a more bluish, this is more purple. And again, by doing image analysis on a mobile phone with principal component analysis to cluster the data, we can clearly distinguish which one of these viruses causing the infections. So where do we think that rapid antigen diagnostics fit in the overall landscape of testing? And so I'd like to mention just some of the advantages, but before doing that, I'll also say that each of these testing modalities, whether it's PCR for nucleic acids or serology or rapid tests, each one of them has applicability in its own test, or rather use case. So this isn't a contest to see which one is the absolute best, but you have to figure out which type of testing you want to do and in what environment to decide which type of test is the best for that setting. So in terms of the rapid tests, these are tests that are equally suited for using in affluent and metropolitan areas, but importantly, they can also be used in rural areas and in places in the world that are under-resourced. These are very inexpensive tests, they're very simple, and can have applicability around the world. They also don't require special uses, or rather, they don't require special equipment or training or chemicals to use, and they can be scaled up quite quickly to make them available to large numbers of people. So what is, from a research side, where are we going with this kind of technology now? The first bullet point here is one that we consider very important and is a major interest in the lab. And that is, how do we anticipate the next epidemic? And hopefully that won't be a pandemic, but how do we develop tests before they become a countrywide or worldwide problem? And there are ways of doing this, we believe, with doing controlling costs at the same time. But by working with field biologists and ecologists, there are hints of data that can be directed, or rather collected, to tell us which of these viruses might be the next threat. And knowing which virus, or having hints about which one might be next, we can then work to proactively develop these diagnostics, because now the response is always reactive. We don't think about developing the diagnostics until it's after a problem has arisen. And that was acutely obvious with the COVID problem, the SARS-2 virus, where there were no diagnostics. Another effort that we're making is we'd like this technology to be possible for local manufacturing, so that they could not only be used throughout the world, but even manufactured throughout the world. And by having simple tests that can be made locally. Part of the need for doing this is to decrease the cost of antibody discovery. That's probably the most expensive part of the process. And we're working on ways to develop new antibody uses at lower costs. And then finally, we have to be able to manufacture at scale to be able to produce these so that they're available to people who need them. So I'd like to acknowledge the people who have done the key work on this. Irene Bosch, as former member of the lab, she's now at E25 Bio. Elena Depuch is a graduate student, and our funding from the NIH. Thank you.
Thank you so much, Brian, and thank you so much to the MIT SENSE Committee for including me here in the program. And I'm thrilled to share some of our recent work in the area of GI device development, but then also introduce how we start to link some of these systems into sensing and micro nanofabrication technology. So with that, let me kick off with a very well-known quote by a former Surgeon General. This is C. Everett Koop, who was a Surgeon General here in the US a few decades ago. And he's known for several things, but he is certainly known for the quote that's included here that, I think we recognize as, obvious and simply, put that drugs don't work if people don't take them. And this really represents the core areas that our group focuses on. And with a particular focus on thinking about how we might make it easier for patients who need to take medications like insulin or biologics through alternative routes, but specifically through the oral route. And as we sort of consider these challenges, we've been exploring a whole range of methods on doing this over the past 10 years, and I'd like to share some of that work with you today. And I wanted to start off by sharing with you some details of a couple of patients that I looked after about 10 years ago. These have been anonymized, but nevertheless, I think really help capture some of the elements, the key elements that inform some of the technologies that we continue to work on to this day. So the first case is a gentleman that I met approximately 10 years ago when I was doing my fellowship at the Mass General, and I remember being called to the emergency department with a request to evaluate someone who had been observed to ingest shards of glass. And this was someone who had been transferred from a local jail. And, I walked down to the emergency department and, I'll never forget, as I walked up to the patient, what I could see was that this gentleman was sitting in a chair. He actually was wearing handcuffs, but in his hands, he also had a sandwich that was half eaten. And so someone in the emergency department had deemed, wanted to help and had given him a sandwich. But of course, that precluded me as the gastroenterologist in trying to help this individual, this patient, with an intervention, just because having eaten, that increases the risk of things, coming up and going down the wrong way. But really taught me a fascinating lesson that has helped inform a significant portion of our work. And that is the incredible capacity that the GI tract has to tolerate sharp objects. So in this case, we actually observed this gentleman over the course of a weekend. The shards of glass were actually about below 2 centimeters, which has been recognized to be a threshold where we tend not to see or have much lower risk of damage in the GI tract. And subsequently, I did quite a bit of reading and then also subsequent grand round presentations around this area and learned a tremendous amount around the tolerance, essentially, of the GI tract. And one of the things I learned, I'm not suggesting folks here on the call do this, but if a room full of folks were to ingest, for example, a razor blade, 90% of folks would actually do fine. Again, it's not something I'm suggesting, but just I think gives you a sense on the the incredible capacity that the GI tract has And certainly the risk goes down tremendously as the sharp object, or the size of the sharp object, decreases. Now, a more common case that we tend to see, this is another patient from about 10 years ago now. It's a lady in her 60s who suffers from osteoarthritis and has been taking a lot of ibuprofen for her joint pain, and comes in essentially with complaints that suggest a potential gastric ulcer. In this case, she described discomfort or pain in her belly. Her stools were dark, often described as black and tarry, and she was feeling lightheaded. And so we performed an endoscopy. And when we see an ulcer, one of the things that we do is we inject epinephrine or adrenaline. And the epinephrine is injected really for its local effects. And by that I mean what we want is the drug to help constrict or stop the bleeding so that we can get a good look at the ulcer and then put clips or cautery to really help treat that. But what we recognize is that as soon as we give that injection, behind us or next to us where we have our heart rate monitors, they start to, they start bing-bonging. So we recognize that although we injected that drug for a local effect, it also has systemic effects. And so that drug that is injected into the wall of the stomach, within a few seconds actually is having an effect on the heart. And so putting these pieces together really motivated some early concepts around ingestible injector systems. And so what you see here are some of the earliest CAD drawings and some of the earliest prototypes that we built in the lab, and also the subsequent testing in pigs. And what we recognized was that these systems actually could pass safely through the GI tracts of pigs. And when we injected insulin, what we could do was actually lower the glucose level to a comparable level, even greater and faster than when injected. And so this really motivated us to continue down this path to try and see how we might develop systems that enable the delivery of drugs like insulin orally. And so about 6-7 years ago, where we were was we had this challenge in front of us. We knew that we could deliver medications like insulin orally using devices that, for example, used injector systems, but there were 3 areas that we really needed to address. And one was, how do we get a device to interact or engage the mucosa in a predictable and reliable fashion? Number 2, how do we get that device to autonomously trigger to inject? And then number 3, how do we get a clinically meaningful amount of drug delivered into the tissue? And so let me walk you through what we did. We were inspired by the leopard tortoises as well as weeble wobbles on developing self-orienting systems. And using these systems, what we found was that by testing these systems with high-speed photography, but then also in vivo, we found that we could identify mathematically, but then also experimentally, optimal shapes and density distributions that could provide that reliable and predictable interaction between the device and the tissue. And as I said, we demonstrated this in vitro with high-speed photography. We also tested this in pigs, where we looked at the effects of movement, for example, on these systems and their capacity to self-right. We also looked at self-orientation in different fluids to simulate self-orientation in the gastric environment, which is often full of fluid, or not full, but at least there's a significant amount of fluid in which the system would have to self-orient. So this was sort of the first challenge. But then for the next challenge, what we had to identify was a cue, a universal cue that we could then apply towards the triggering. And the cue that we honed in on was humidity. And the gastric environment is a humid environment. And so therefore that, made us think about and start to consider different materials perhaps that could be sensitive to humidity. And what we wanted to do was inject some kind of drug formulation relatively quickly. And so we considered, what if we could find a material that could sense humidity and potentially, use it to hold stored energy, let's say in the form of a compressed spring. And, and so what we wanted was a system that could release that energy all of a sudden. So ideally, what we were looking for was a material that displayed brittle fracture mechanics. So I want you to think candy cane, not taffy. And what you see here is one of those early prototypes where we encased a solid spring with sugar glass. And you see that rapid release that enables the rapid injection of a drug formulation. And we were able to demonstrate through simulation, but then also experimentally, that we could tune the time of actuation by controlling the diameter of the sugar cylinder. Now, we've addressed number 1 and number 2 in the challenge list. And so number 3 was, how do we deliver a clinically meaningful level of drug? And one significant boundary condition here was that we wanted systems that were the size of a blueberry. And so we wanted that for safety. And so what we did was actually we shifted the drug formulation to maximize the density of the drug. And so we shifted, typically these drugs are in a liquid formulation, but we shifted to solid formulations. And we generated millineedles that contained anywhere from 80 to 100% of the active pharmaceutical ingredient, in this case, insulin. And so what we have here on the bottom right is one of those early prototypes with that pure insulin, in this case, tip. And so putting this together, we evaluated these systems in pigs. We showed that we could deliver a significant amount of drug that was comparable to the levels that would be achieved when injected through the skin. And so essentially, again, what we have is a system that is able to self-orient, sense the humidity, trigger that injection event, and then have that solid formulation dissolve in the tissue to exert its activity. So you might be asking yourself, well, how does this relate to sensing? And this is some more recent work that was just published just a few weeks ago. So one of the things that is really critical, and we really have been working very actively in the lab, is thinking about how we can inform the patient, but also providers with whether patients are taking the medications as prescribed to really help that the patients maximize their health. And so with that, we've been exploring the incorporation of electronics into a whole range of different drug delivery systems. And so one of the things that we've been exploring has been incorporation of electronics for a couple of different aspects. One is using the electronics to exert an effect. So for example, to stimulate the tissue, but potentially using the electronics to sense the position, whether someone has ingested it, and whether there's been engagement with the tissue to start to inform device function inside of the body. And so what we did here, using that self-orienting platform, initially we added some batteries. You can see some early prototypes here, but we showed that using these systems, we could actually stimulate the tissue to exert a muscular contraction. And so, and then beyond that, what we've also explored has been actually using the actual sensing capabilities of these systems by having a patch on the surface. And what we can see is essentially a difference in the signal of whether the device is in fluid or in contact with the tissue, or of course, if it's been excreted from the stomach. And we see very distinct differences based on the signals. But also what we're able to do is communicate from the external patch to the device. And so in future, some of the things we're looking at are external triggering of an event. For example, delivery of a specific drug that is coordinated by the external patch. And so, where we are now, what we recognize is we have a set of tools to really, we hope will help many people to deliver a broad range of drugs, including biologics like insulin, but also others. And certainly from the drug delivery perspective, we're exploring a whole range of different drugs. On the sensor side, we see applications on adherence or compliance monitoring, but also in the performance monitoring of the device, as well as sensing, whether it be pathologic or physiologic signals. And additionally, we continue to expand the capabilities to really inform potential ways of having these systems stay in the body for a longer period, potentially harvest energy if they are staying for a longer period, so they can sustain communication and function. And then, as I mentioned, starting to think about the external triggering. And so with that, it's my pleasure to present the work of a village. And the person in blue over here is Alex Abramson, that led a lot of this work in collaboration with some incredible collaborators, both locally at MIT, but internationally, including the Novo Nordisk team in Denmark, as well as the Gates Foundation. And here's our team in a socially distant outing, who I also want to thank for their incredible support and tremendous efforts. With that, here is my email. I know we have a Q&A session at the end of our section, but just in case, if there are any other questions, please feel free to reach out. Thank you so much.
Hi everyone, this is Canan from MIT Media Lab, and I direct a research group called Conformable Decoders. We believe that we live in an ocean of physical patterns, heartbeats, respiration, muscle movements, and temperature change, and so on. And we think that these physical patterns contain information that needs to be decoded. And in our group, we fabricate flexible, stretchable, conformable decoders, devices, which can have intimate integration with the body parts such as brain, GI, skin, and so forth. And we explore the human body in a systemic level from various types of human parts with implantable, wearable, and attachable devices. And we do all this conformable devices in our mini version of MIT.nano. It's a clean room, first of its kind clean room at the MIT Media Lab, and we call it yellow box because literally it's a yellow color because of the UV sensitive polymers that we use, and we made it physically transparent so that anyone can see whatever we do inside the clean room without taking any permissions or trainings. For the scope of this talk, I am going to introduce you an attachable device, which is literally a personal garment that you can wear intimately on your skin. And this device can do real-time, continuous monitoring of the body's signals like respiration, heart rate, and temperature change relatively on the top of your body part, like the upper level of your body. Of course, there are many great researchers all around the world who works on skin electronics, as well as textile electronics in different versions. For instance, the skin on electronics enhance mobility, but they are mostly fragile and not perfectly scalable, and sometimes inappropriate for dynamic activities. Whereas textile electronics is a good option for large-scale sensing and can withstand with dynamic activity, but mostly it is expensive and unconformal. So in our technology, we try to sort of combine this on-skin electronics with textile electronics in a way that your electronic systems will use the textile as a backbone, as a substrate, and you will wear the t-shirt and nobody will understand you have electronics. But at the same time, you will be able to track your vital signs such as temperature or moisture or acceleration. For textile electronics, mostly people use the electronic yarns or conductive traces, whereas these technologies are mostly very susceptible to humidity or temperature change and washability, and not sustainable for the washability. In our technology, we provide a solution which is called Electronics Textile Conformable Suite, which I will refer E-TeCS from now on. And it's a multi-model sensor network on a conformable textile. Again, we use the textile as a passive form, as a passive substrate, just to hold our electronic components in a continuous manner. The features of our technology is that it's large scale. We can cover the entire body with no discontinuity, and it can do multi-modal sensing, temperature, heart rate, respiration, and many, and maybe many more in coming future. And we can use them in daily life. Like they are very durable, they are continuous. We can use them in a dynamic conditions, such as while we are doing gym activities. And of course, most importantly, the device, the textile platform is stretchable and flexible, and we can tailor it for your body parts. It can vary from a premature baby body to an adult body, an older person body, without any problem. And how we do it. This is a small, tiny video that I would like to show to highlight our technology, and so that you can have a brief picture about the project. And as I said, this is an electronic textile conformable, and it's tailored. We can change the shape and the area of the electronics with no as I said, this technology is scalable. We can simply see the different types of electronic connection of electronics with a simple knitting technology, and we can have the shape of your body. And this is the wirelessly controllable system that we can collect the data. Okay, I lowered down the sound. Looks like you can't hear me. Sorry about that. So this device is also washable, that we can detach the wireless module and place the device in a washing machine and see the temperature change and acceleration depending on the modes that we use. And we have in total 30 skin temperature sensors, as well as acceleration that we use for the heart rate and respiration to track the physical activity detection, especially when we do the gym activities. This is one of the real-time sensing during the physical exercise in the gym, where in real time, we can track the temperature change on human body, as well as we can see the visible heartbeat and respiration throughout the entire exercise with an associated skin temperature heat map. And now in a moment, you will see the acceleration will increase when the person starts running. And at the same time, temperature will slightly decrease because of the sweating that occurs on the human body. And of course, we can use this technology in many other applications, but this is one of the trials that we tried on a healthy subject with an age of 25. And when you slow down, again, the acceleration decrease, heartbeat and respiration decrease based on the activities that we did. And how we do these devices. Now I would like to talk a little bit about the design. So we use off-the-shelf electronics and encapsulate them in a biocompatible polymer and use in textile electronics and create housing through the digital knitting so that we can encase this electronics and have an open window for this electronics to have an intimate integration with the skin. And we also use thermoplastic polyurethane shelf on top of the devices so that the device can withstand to washing repeatedly in the washing machine with no electrical and mechanical degradation. We also have islands and stretchable bridge structures where we have the electronics on islands, and the interconnection, the copper lines provides us the electronic interconnections with 30% elongation. And we can also use a commercially available flexible board system where we can fit a total of 66 temperature sensors and 20 interconnection systems, which can easily demonstrate the large scale and rapid manufacturing of this approach, which makes our devices extremely cheap and deployable on large scale. And for the digital knitting part, or the textile part, where we use two different structures. In one is a programmable interface, the digital mapping. It's because it's programmable, and it can give us automatic machine processing of stitching of the interlocked loops for multiple strands of yards. And we can also use interlocking loop for different types of parts for the textile electronics, such as textile channel, as well as the interlocked stripe, where we encase the electronics inside, house the electronics inside with no mechanical, again, and electrical degradation. And we used 100% high flex polyester fabric, and with a detachable processor, microprocessor, and Bluetooth system, along with battery, where we can charge the device with no problem, so that we can detach it from the textile electronics and charge it, as well as we can wash the electronics, or wash the textile without the module attached to it. And this will also allow us to real-time, simultaneously collect all the data and process and have an understandable signals, kind of an interface between medical doctors and the patients. And in terms of fabrication, we use 2-layer of industrial flexible PCB boards, where we use the soldering serpentine integrations for the islands where the off-shelf electronics are on top. We also use epoxy resin for encapsulation, and the polyurethane lamination for the hot air gun. And after this manufacturing, then we use the textile to embed the electronics inside, as you can see from here. But to do so, having the textile is one thing, then we take the measure of your body and create front part, back part, as well as the arm part. Then we stitch all of them together so that it will fit into your body. It will be tailored and it will be personalized. And then later on, we can house the stretchable long strips of electronics into the textile with no physical appearance from outside. And all of the sensors going horizontally throughout the stripes are collected with 4 thin copper wires vertically, and they can all go to the main hub through the IC2 protocol. With this system, the main module contains a battery as well as a microprocessor, and also Bluetooth system, so that we can collect the data from all the sensors throughout the body and process and create a colorful map, so that we can understand what's happening on our body in terms of the temperature change, heart rate, and respiration. And of course, we do characterization, we do electromechanical characterization, stretching, washability tests. We also do body activity and sensing modalities. Our device can be stretched up to 30% with no electrical and mechanical degradation, and all with the force enforcement, the change in the resistive is really so tiny, which suggests that the device can be used up to thousands of cycles with no mechanical and electrical degradation, which is good because we want to use this in our daily activities. We also show the washability test of our devices where the electronics are integrated on top of the textile, and to show that we use an LED system and sort of wash the system in multiple washing cycles and check the intensity of the LED to show that the electronic degradation is actually not happening and it's very negligible. We also do the washing test and see the temperature change when we have the cold water in the washing machine. We can easily see that temperature drops, and we, for instance, spin it, the acceleration increase as well as temperature is increasing. We can also show such changes with the device that we have. So later on, we also show the temperature accuracy of our device and sort of cross-validated with an IR camera. And you can see from this top image, it's an IR image where we have the electronics underneath on the hot plate, and we increase the hot plate up to 34 degrees Celsius, and we can find our sensor accuracy almost 0.1 centigrade Celsius and precision of 0.01 centigrade Celsius. And we also did FEM simulation to show that experimental results capture well with the theoretical results. We also show the mechanoacoustic response of the accelerometer embedded in a fabric and simultaneous cross-validation with the EEG sensors, where we can show the respiration waveform after eliminating the low-pass filter of the high frequency from the heart. And you can see with the close magnified images from here, all the signals, again, there is no time for these details, but all the signals actually tells about our heart rate and respiration and how the ventricle and atrium of the heart contract and relax throughout the exercise. As a conclusion, our technology can provide a large-scale, multimodal, low-cost, and mass manufacturable structure for long-term use, and it is modular and it's tailored, and as well as it's very comfortable and conformal. Of course, beyond of this experiment, we would like to increase the battery life of our technology, which is currently 5 hours with all the sensors are working. And we would like to increase the sensor modalities, and also do want to do more electromechanical optimizations. And the applications are almost limitless. We would like to use this in healthcare, as well as telemedicine, sports applications, and space exploration. And this is my team, how we look at in our clean room. And with this, I would like to thank our funding agencies, NASA and Trish, and thank you for your attention.
Amongst the reasons for being admitted to the hospital, the third most common is dehydration. At the same time, millions of people in the United States suffer from too much water. These are people like congestive heart failure patients or renal patients. Many physicians would tell you if there was a convenient way to make a clinically relevant measurement of hydration status, it would be a vital sign, just like temperature and blood pressure. So we don't have that yet, and this talk is about maybe one of them. I won't get through the details of this today, but there is a paper out that came out last year that basically summarizes the findings that, or goes in more detail of the summary that I'll present. But we made a number of discoveries in this paper. What we were doing was using a type of magnetic relaxation to characterize water in the body. And we discovered that the interstitial water in muscle, skeletal muscle in particular, acts as a key reservoir for fluid in the body. And in fact, it can tell you with an absolute scale of whether you have too much water or are euvolemic, as we say, normal amounts of water. And in this study, while we used an MRI as sort of the gold standard, we did a bedside tool that uses the same basic principle and showed we can get the same type of measurement. There are many different ways that people have tried to measure hydration status. And if you go to the web today, you'll find many of these. But unfortunately, after decades of attempts, none of these is proven to be clinically relevant. So few, if any, are used in the clinic. And mostly what is used in the clinic is qualitative and subjective. Probably the most common that many of you are familiar with is this lower extremity edema. This is for people that are hypervolemic, too much water, their kidneys haven't functioned properly. And in the extreme case, the fluid begins to pool in their ankles, and you can literally push on their skin and see the fluid move around. That's way beyond what should be allowed. We need to detect the presence of too much water way before there's obvious symptoms like that. We chose for our study a group of patients that were going to have a large volume change in a short period of time, specifically hemodialysis patients that go into a clinic and basically they're hooked up to an artificial kidney that does two things: removes the toxins and also does what's called ultrafiltration, removes the excess fluid. And then depending on the patient, this can be several liters of fluid in a 4-hour session. And we thought this might be an ideal situation where we could do a measurement before and after dialysis and actually know how much water was removed in between, because we actually measured it, and see how that's reflected in our measurement. It also is a very important economic setting. Just the hemodialysis space costs the US around $42 billion a year. This is a very big thing, and affects almost 1 million patients in the United States. So this is a big deal, and there's a lot of morbidity associated with this. And these folks spend about 3 days a week dealing with their disease, going to the clinic. They may be only 4 hours in dialysis, but they got to get there and come back. And as a result, they spend 3 days a week doing dialysis, and they will the rest of their lives. So what about MRI that does this? Or more specifically, what about NMR that tells us about this? Well, NMR and MRI, as normally practiced, are done with spins or protons that are made up in water. So the MRI that are composed in water. So MRI, for example, is mostly carried out looking at the properties of water from one tissue to another. And the property that we're mostly interested in, or what it measures, is the relaxation behavior of those protons. So you sit in a magnetic field, the protons align, you send in an RF pulse, excite them so that they're no longer aligned with the field, and then watch them relax back to their aligned position. And that relaxation time is characteristic of the tissue the water is in. And the amplitude of the signal is a characteristic of how many protons are there. So the combination of those two things, we can measure the combination of those two things with an MRI, or more specifically, an NMR measurement. And our hypothesis was we could do this quantitative type MRI as a function of position in the body and characterize how much water was there and what type of water it was. The study design is pretty simple. We would do an MRI scan before and after dialysis, and we would also do blood draws and other types of measurements and watch what happens before and after dialysis. And we also did healthy controls to compare with this. The MRI protocol, we focused on the leg, specifically the calf, and we could basically measure the NMR properties of every muscle in the calf. And we modeled each point in the MRI as a exponential decay. For reasons that we'll go into in the paper, those, we know that there are two types of relaxations at every point. And we mathematically can extract both the T2 for each one of those relaxations and the amplitude. And if you do this for regions of interest that include muscle and subcutaneous tissue, this is the histogram of relaxation times that you get. And you can see there's two populations in each type of tissue. One relaxation is very similar between muscle and subcutaneous tissue, and the longer relaxation is different in each one of these tissues. What we know is physically, the longer the relaxation time, the more free the water is. So it's saying that water in the subcutaneous space is very free to move. That in the muscle is less free to move. The reason why there are these two types of water in each compartment, each muscle and subcutaneous space, is that there's water inside the cell and outside the cell. This intracellular fluid relaxes very fast. The extracellular fluid outside the cell takes a longer time to relax because it's freer. And this extra water here turns out is the reservoir. When you're pulling water out of the blood in hemodialysis, it actually comes from the skeletal muscle. We discovered that in this study. So here's the amplitudes of this long relaxation in the calves of healthy controls and dialysis patients. And I think you can see here that there's more yellow, longer, and yellow and red in the dialysis patients than in the healthy control. That's just a graphical instance. If you look at the histogram of this amplitude of long amplitude in muscular tissue in the dialysis patients before and after, so AM and PM, what you see is in every case, the amplitudes get smaller as you go through the dialysis process. In healthy controls, with 2 exceptions that I'll return to in a moment, there's no change. So what's with these 2 healthy controls? Why did they decrease? Well, here's the cumulative distribution function. Is basically the integral of what you saw in those histograms for both dialysis patients, the dotted lines, and the healthy people in the AM and the PM. And the difference between these is going to be the difference associated with dialysis. So if I take the same patient in the PM and subtract the AM curve, I'll get a delta. Whoops, sorry, skipping ahead. And here's the delta. The first thing you can see is down here, the solid lines are the healthy controls. They don't change except for those 2 exceptions. And the dialysis patients all decrease. They get less water. They had removed water, so the amplitude could decrease. So what about these 2 healthy controls? Well, it turns out if you just lay in a gurney like these healthy patients do and don't drink, you dehydrate, you lose water. And you could tell that from their blood chemistries. They basically got dehydrated just sitting there because they didn't drink. One thing about being older, you lose your sense of thirst, and that's why it's so much easier to get dehydrated when you're old, speaking from experience. So the most important thing that came out of this, though, is that if you look at the amplitude of this long relaxation time, you actually can tell hemodialysis patients from healthy controls in the first measurement with statistical significance. This is an absolute measure of hydration status. And of course, you saw the difference here. So this thing told us we were really onto something, because now quantitative NMR can actually be shown to tell sick people from healthy people with a single measurement. Now, how do we translate this? So in the last minute or so, last couple minutes, I'll tell you how we're translating this. We don't need an MRI. What we did in this study was to also build an ex situ NMR. So basically, these are rare earth magnets cobbled together into this array we call a sensor here, with the little RF coil on the top. And that's pushed up against the calf while this patient is in bed. And it senses a region several millimeters away from the surface of the magnet in the tissue. And it can be from 2 to 5 millimeters. And so that captures it, like you can see in this MRI here, we've sort of labeled in red the small volume that's there. Now in this particular sensor, it captures both muscle and subcutaneous tissues. So we have to do signal processing to extract it. Here's just another view of the sensor. This just shows a comparison between the MRI relaxation time for a phantom with known relaxation time compared with our sensor. You can see it's pretty good. And here's the delta using the sensor. So hemodialysis patients and healthy controls, you can still see that we can measure the change. However, the biggest problem we have in that particular sensor is that it was designed assuming that the excess fluid that we were removing was in the subcutaneous space, because that's what everybody thought. Instead, it captures both the subcutaneous and the muscle in this particular design of this sensor. And so you don't get an absolute measure with this sensor. But if we had done 2, we can use the MRI data to say, look, if we had had the sensor right out here, you'd see that you could get a statistical significance in the change just by that single measurement. So this is the new sensor. First off, the new sensor will have shape pulses that actually moves the sensitive region from this distance to deeper into the tissue. And the second thing we're doing is changing the array of magnets so that we go from a volume that looks like this, sensitive volume that looks like that, to one that looks like this. And that does two things. Not only does it get it deeper, but because the volume's larger, we get a huge increase in signal, which means we don't have to acquire very long. So we think we can do this under a minute. This is my last slide, or actually second to last slide. I have an acknowledgement slide. Is basically where we're going to take this. Why would a clinician use this? Other than to say, well, you know, they can tell it's a dialysis patient. They already know that, right? Just by the first measurement. But what they could tell is how close are they to the dry weight? How far away are they from the correct amount of water, the euvolemic state? So they can actually write a script saying, take this much fluid away. The second thing we want to do is look to see, does our measurement predict the adverse events that happen during dialysis, specifically cramping and hypotension? People pass out all the time because they are withdrawing a fluid too fast. Could we tell before those AEs happen that we're going to have that? And then finally, all these things are all diagnostics. We want to compare against quality of life measurements. Do people that do less well on dialysis actually reflect that in their measurement? They continuously are higher volume than they should be. And there's my last slide of acknowledgements. So we had a lot of great funding sources, and I would particularly acknowledge the MIT MGH Grand Challenges grant, which funded the clinical trial. Thanks.
Thank you, Lee, Giovanni, Canan, and Michael, for those wonderful presentations. I'm now giving you a little bit of time to get from your desk back to your desk to join us at the panel. Let me give you the answer to the trivia questions, or the history questions. So the first practical electrocardiogram, or the ECG or EKG, depending on where you are in the world, was in 1895. As you see, we've progressed quite a ways from having to stick our arm and our leg into a bucket of salty water to characterize the electrical signals coming from the heart. That was in 1895 when that was first put into place. The first full body CT, computed tomography scanner for the full body, came about in 1973, so not that long ago. And on the blood oxygenation, the pulse ox, the technique to use two different wavelengths of light to characterize the relative absorption of oxygenated and deoxygenated blood, was invented in 1972 in Japan. The first device practiced on patients was 1975 on the ear. That progressed then to wearing it on the finger, and now sort of modern day on the Apple Watch, but only was really commercially available in 1980s, despite the fact that the extreme prevalence of this as a biomarker, as a signal, that's used readily in the hospital. So I think with that, we have our colleagues joining us in the breakout room. Very good. Lee, Giovanni, Canan, Michael, good to see you. Thank you for your talks. As I'm hearing here, we have a bunch of colleagues from industry, both manufacturing industries and pharmaceutical industries. And I'm struck by two common themes. And so the first common theme that I would like to ask you about is in the realm of manufacturing and manufacturing challenges, and recognizing you're all at various points along sort of this translation from research into something deployable. But I want to ask sort of a multi-part question, that if you had a go signal now and you needed to make 1,000 of your sensors or of your devices, how long would that take? What are the challenges to be able to make that 1,000, for it to be repeatable 1,000? And in that answer, what is the role that you want to play as a researcher versus the opportunity and role to collaborate with manufacturers that may be more interested in figuring out how to deal with those issues of making 1,000. So I'm gonna go with, I'll pick on Lee first to start, because he specifically had manufacturing as one of the as challenges of ongoing work, while we give everybody else the chance to reflect.
Thanks, Brian. So we've already made many more than 1,000 of the tests, and part of that is due to the fact that lateral flow chromatography for these rapid tests is a very well-established technology. It's been used for decades, and fortunately, there are many companies around. We're using a local manufacturer that can make these tests and at a very large scale. There's one company in the UK, for example, who has told us that if they scaled up to full capacity, they can make 8 million tests per day. So it's huge numbers. The challenge for us is to have a manufacturer that's willing to work with us, because whenever everyone here knows that whenever you go from a laboratory device to scale up, there are always things that you have to worry about, because not everything scales perfectly. So having a manufacturer that is interested in this type of technology and is willing to work with us to make sure that there aren't false positives and background and things like that would be very important parameters for us. Finally, we're also interested in facilitating manufacturing, not only in the US, but also in other countries, particularly low-resourced companies where they need to make these devices locally and at a price point that they can use.
Thank you, Lee. I'll take a volunteer, or I'll pick on Gio next, going in the order that.
No, thanks so much, Brian. And you know, to the manufacturing question, I mean, it's certainly something that we think a lot of. We spend a lot of time thinking about, but it really depends, you know, on the project. And so, the systems that I presented today were, you know, have been developed in collaboration with Novo Nordisk. And so, part of the development, certainly, we didn't know where we would end up or what would work. And so, that really early stage work, you know, is very much, I would say, you know, in general, what we focus on in the lab. But certainly in that context and that specific sort of program, you know, once we could see a potential path forward as far as devices for the oral delivery of biologics, what our, you know, collaborators and partners really were helping alongside was actually, and pretty early on, even before the initial publications, we already had plans that we had developed together on how to make 1 billion of these devices annually, and you know, to a significant level of detail on everything from the actual manufacturing processes all the way to, you know, waste impact analysis and how you know, how this would impact, for example, Denmark, and really thinking through all of those steps. So you know, that I think is one example where, you know, we started to de-risk some of those fundamental early concepts in the lab, but then as, you know, we progressed, the manufacturing risk was really mitigated in collaboration with, you know, a major partner. There are other situations in which the manufacturing sort of constraints or boundary conditions are brought in even earlier. And so I'll give you a more recent example. We've been focused in setting up COVID on making masks. And one of the things that we constrained ourselves very early on was on using systems that we recognize as scalable. And so we really constrained our development and design around injection molding. And so, you know, that constrained the materials that we would use and some of the processes. So, you know, it certainly depends on the projects, but often, you know, if we're developing drug delivery systems or ingestible sensor or implantable sensor systems, the risk is more on the fundamentals of the engineering or the science. And certainly we work with different partners, but there are projects, again, where the manufacturing constraints are built in even earlier because we need a path forward that can be amplified very quickly. So hopefully that gives you a little bit of a sense on at least how we approach that.
Thank you. Shannon, I'll take you in the next speaker order here.
Sure, thank you, Brian. I will echo Lee and Gio by saying that collaboration at an early on is very important, yes. And the project that I highlighted today was actually kind of an example for that. MIT Media Lab had a collaboration with China, and they could send students to Shenzhen electronic market. And I personally benefited from this program and sent one of my students to China last summer for this textile project. And we work very closely with the manufacturers. And we learn what their interests are, and they learn how we approach the problem. So it was kind of a mutual discussion along the way, and it was really helpful for us as a researchers to see how the manufacturers are designing their frame of work, and how sometimes we have difficulty to understand each other, because sometimes our timelines are very different and our interests are very different. We mostly would like to file a patent and write a research paper, whereas they really care about the deployment and large scale and so forth. And the devices that we built here cost us like hundreds of dollars, whereas in China it was just a few dollars. And it's because we use the labor, local labor and local materials. So it was really, really helpful for us to understand how the large scale will benefit our research, because right after that initial preliminary research, we wrote a research grant to NASA and got a grant seed funding, which allowed us to kind of carry our research to the next level. And another thing is that I think such programs in departments, in research institutions are really helpful. A very recent example, I started collaborating with the Dean of Engineering, Ananta, and we started a collaboration with Texas Instruments. And again, at some point you do your work as a researcher, but when you talk to these manufacturers, you learn more from them. You save time and you save materials, because most of the time they send you materials and sensors, and you learn from them quite a lot, which makes you to progress faster, as well as learn how to do large scale manufacturing. Another example that we started in collaboration with a research institute in Singapore. For instance, in our clean room, we only use 4-inch wafer processes. With the collaboration, we learned how to do 8-inch wafer processing, and it saved quite a lot of time and money. One of our research will be online in a few weeks, and you will hear about it. It's a stretchable sensor that you can laminate on skin, and it can make it to nonverbal communication, especially for ALS patients. And one device that we created, it's perfectly disposable, is just $2, due to the fact that we can do 8-inch wafer processing with the manufacturers that we have as a collaborator. So as a young researcher, I benefited a lot from those programs, and I hope this would be in other research institutions as well, because I believe it's giving a lot of guidance to the young researchers especially.
Thank you. And Michael, I know you have a lot of experience in this domain, so, but in particular related to the sensor that you're trying to,
Yeah, I'm a bit lucky in that regard, in that, you know, the first small relaxometers I built ended up in T2 Biosystems deck. So, you know, they're a diagnostics company, molecular diagnostics company, and every one of their instruments has a relaxometer in it. And, you know, fundamentally, well, it's not an ex situ sensor like I described. Fundamentally, the parts and everything are very similar. So I know it can be manufactured. If I were doing it, as I hope to do again with this technology, I would outsource as much as I possibly can. As long as the supplier can you know, meet the requirements of your quality system, because we're making a medical product here, it makes so much more sense to work with a manufacturing partner, and for at least the large subsystems like that. Actually, the biggest limitation for this technology, I've already been asked, because over the past year since the publication, I've had a number of companies approach me. I actually think the rate limiting step is not the clinical, It's actually the user interface. You know, nurses and physicians are going to have to use this instrument. And I've learned that don't underestimate how long it takes to make a screen that the FDA will approve to be in front of a nurse. And where that is going to make a decision about a patient based on what they see on that screen. So I think I actually don't think it's the manufacturing so much as the design of things like the user interface that are a big impediment to actually, rolling this technology out.
Thank you for that. That actually brings me to the second question that I will also ask of the four of you, and I will go in reverse because Michael already started to answer it. But in this area of, I'll call it sort of closed loop usage of the sensor, and we have a lot of visitors here from the pharmaceutical industries and there's increasing opportunity to do controlled drug titration through sort of measuring what's happening in the body and maybe delivering dosage based on that. But what are the sort of the barriers to doing that, either for deciding a treatment of care, whether it be a pharmaceutical or not? And I'll go with you, Michael, to start.
Well, yeah, well, and I sort of alluded to the example of congestive heart failure. All those patients are on diuretics, and the right dose varies. So you got 5 million congestive heart failure patients. They cost us 6 million hospital days a year. The majority of those hospital days is associated with drying them out. Basically, they have to go on IV diuretics. And it's because they weren't taking their oral meds correctly, or even if they were, the right dose the dosage should have been changed. So if they had a measurement that they could do at home, you'd know exactly where they were in the volume status range, and you could dose diuretics on it. In the development of new diuretics, I mean, that would be having this type of biomarker for stratifying patients or doing, so it's not just in the use, but even in doing clinical trials, identifying patients that, the phenotype that you want to study based on their volume status. Now it would be possible to do. There's never been anything like that. And in heart failure, there's all kinds of patients. And so what you want to do is actually select out specific phenotypes to study. So that's just, I'm sure that other speakers have other examples.
Thank you. Anybody want to pick up on that? Reverse order if we need to voluntold.
Okay, I'll go maybe. The closed loop is important, and I would actually give two examples. One coming from a paper with Michael Cima, and another one, a paper with Gio that we wrote together. One of the devices that we developed is an implantable device which can go inside the deep brain and infuse multiple drugs on demand while reading neuronal potential, so that we don't blindly just infuse drugs. We actually read out the neuronal potential and infuse the drug accordingly, which is kind of a closed loop system. This is important because you want to test how your brain is doing while you're infusing drugs. You also want to know how your drugs are affecting your brain neuronal activity. So this is one example. And another thing that we did with Gio is an ingestible device which can go in the stomach, and we self-powered with the motility in your GI tract, but at the same time can be housed for multiple sensors to read out the temperature, pH, and so forth. And this is important. Think about in a systemic level. What if we have this GI tract, but at the same time implantable device inside the brain? While you are checking the motility in your GI tract, you may also would like to read out the neuronal potential in your brain. It's a step beyond, not only the closed loop system with two systems, it's also you can communicate these two organs together that was not possible in the future. And in addition to closed loop, I realized that large data collection is also important, because you can, with a simple and very limited data collection, you can estimate what will happen in coming days to your GI tract or your brain. For instance, one of the devices that we generate in our lab is a patch that you can laminate on your knee, and it can measure the quality of your gait, your walking. And we collect lots of data. It's again a closed loop system. We collect a lot of data, And we do machine learning so that we can understand, especially if you are a Parkinson's patient, when you take the drugs orally or intravenously, there is no way that you can track these patients at home. You take the drugs and then you go to the doctor after months, and the doctors communicate with you to ask any symptoms that you experience. But with this kind of technologies, you can generate lots of data, and through machine learning, you can even estimate when your walking would be masked or when you are going to be freezed, and you can know all this information with the help or without the help of the drugs that you take orally. So this is another kind of dimension that I find very helpful, the data collection and processing.
Great, thank you. So Lee or Gio, take the next stop.
And I can add to what both Michael and Canan have mentioned. I mean, I think, there's a tremendous opportunity here to help patients across a range of diseases. And, but when we look at sort of sensor and closed loop systems, like classically, we tend to think about diabetes and sort of glucose sensing or anesthesia, where, there's also closed loop sensing, but really, there's such a tremendous opportunity. I mean, you've heard, congestive heart failure, whether it be neurodegenerative conditions, into really thinking about the fundamental sensor development, but then also the material science that has to go along with it to prevent fouling, maintain the stability of the sensor performance over prolonged periods, depending on where we want the sensor to live, whether it be on the surface, in the interstitial space, or in the GI tract. And so as we sort of think about, what the different use cases, there's sort of, tremendous amount, I think, really of opportunity for us to, as a whole, really to contribute to help patients. But, going back, I think, and considering all of the conditions and, trying to prioritize where can we have a big impact, and, where is it that we're already doing some kind of feedback, but perhaps we haven't closed the loop. And, you can see the evolution of glucose. I mean, the first development of the glucose sensor were in the 60s, and then you could see sort of that evolution over, more than half a century now, and, the incredible impact that they can have. And so I think closed loop sensing is extremely, I think, exciting, but there's still a tremendous amount, as a community, that we need to do across, a range of disciplines to get a few other products over the finish line.
Great. Thank you for that. Lee, in the last minute or so that we have remaining, would you like to add some additional context from your perspective?
Sure. So perhaps one example I could give would be our interest in future work in developing diagnostics for pathogens in a prospective way, not a reactive way like we're doing now. So one way to do that would be to combine simple tests for viruses, other pathogens that we're worried about, in regular testing that patients might be getting. And so while getting normal care, we could be screening, doing surveillance to try to pick up on other pathogens that might be incubating in the background. And that could have a huge impact on readiness. Pathogen readiness is such an important issue.
Very good. So friends, thank you very much, Lee, Giovanni, Canan, and Michael, for the great discussions and the great presentations.